Hi everyone, here in this lecture we are going to talk about the development of placenta. So before we start, I'll suggest you to watch my previous video of ovulation, fertilization and formation of blastocyst so that you will be able to understand how this process begins. So in this part we are going to discuss about the implantation, and in coming parts we'll be discussing about the decidua formation and the functional unit of placenta, that is chorionic villi formation. So before I deal with this implantation, let me draw the diagram first here where this implantation is going to be take place. So the structure is called the uterus. So here it is the uterus, and there it is the uterine tube, and the vagina. Okay, if you remember, we have talked about how the fertilization takes place. So after fertilization, what is happening in the uterine tube is the formation of zygote, and after zygote formation, this zygote move toward the uterine tube and it forms the 16 cell stage structure which is called morula. And after the formation of morula, as it spends 3 days in the uterine tube, this move toward the uterine cavity, and here this morula differentiate itself here by forming a ball like structure, and there it is called the blastocyst. And in the blastocyst there is the outer lining which is called the trophoblast, and the cell which is lying as a group together at one edge, these later forms the embryoblast. So this is how this ball like structure, that is the blastocyst, is formed in this uterine cavity. Now what is the implantation? Implantation means that is the invasion, that is the complete invasion of this ball like structure into this endometrium. As you know that there are the three linings, or there are the three layers of this uterine wall. The outermost is the perimetrium, the middle one is the myometrium, and the innermost is the endometrium. So the invasion of this whole structure is going to be take place in the endometrium itself. In endometrium also there is a two layer. One is the stratum basalis and the another one is stratum functionalis. So the invasion is going to take place in the functional layer. It doesn't invade deeply in the basal layer, or it doesn't go beyond that, that is in the myometrium or the perimetrium. It just invades in the functional layer. So when this implantation occurs, it occurs on the day 6 after the fertilization. And where it does occur, this whole blastocyst invade, it may be on the anterior wall or maybe on the posterior wall. So the implantation is going to be take place on the anterior or maybe on the posterior wall, but near the fundus. So on the 6th day after fertilization, implantation occurs on anterior or maybe on posterior wall near the fundus. So where this process begins, what happened? These inner cell mass send some signal to these peripherally arranged cell, and as this creates some signal, what is happening here? These trophoblast or the trophectoderm differentiate into two layers. What two layers are there? The outer layer, which is multinucleated because there is no exact cell boundaries are there, and there's nucleus and the cytoplasm become mixed together. So this outer lining form the syncytiotrophoblasts, the outer line forms the syncytiotrophoblasts, and the inner line, which is single layer, and there is the exact cell boundaries are there, and this layer forms the cytotrophoblastic layer. So this forms the cytotrophoblast, and the outer line forms syncytiotrophoblast. So by some signals from these inner cell mass, these trophoblasts differentiate into two: outer multinucleated syncytiotrophoblasts, inner mononucleated cytotrophoblast, and this cytotrophoblast also termed as Langerhans layer. So at this time the uterus is in secretory phase, because after ovulation secondary oocyte is released and mature graphene follicle form the corpus luteum, and that releases progesterone, and progesterone increases the vascularity, and also it releases some fluid from the endometrial glands. So uterus in secretory phase, so it receives some nourishment for the initial development of this blastocyst. So the implantation process begins at the embryonic pole. What is embryonic pole? Where this inner cell mass attached with this trophoblastic layer, this pole is called embryonic pole, and the other empty side of this structure is called abembryonic pole. So embryonic pole start moving toward this anterior or maybe toward the posterior wall. So what is happening next is that there are microvilli or the finger-like projections on this endometrial lining, which is called the pinopods, and these pinopods start absorbing the uterine milk from these glands. So before this process begins, it is very important that these pinopods absorb more glycogen or lipid-rich secretion from these glands, so that these are very important for initial nourishment of this blastocyst. So these pinopods absorb more glycogen and lipids, and meanwhile there are some microvilli on these syncytiotrophoblastic cell, and what they all does here is that the apposition stage begins. What is apposition? It means the blastocyst and endometrial lining lies side by side, and the microvilli of both the structure start to come closer, and after absorption of the uterine milk from these pinopod, what is happening? The another stage, that is adhesion, begins. And for the adhesion, there are the adhesion molecules, that is some cadherin, selectin, integrins, and these molecules releases from the both structures, that is the blastocyst as well as the endometrial lining. So the both structure releases some adhesion molecule, and then these microvilli of the syncytiotroblastic cells as well as the endometrial lining start interlocking to each other. They will tightly attach with each other. As this syncytiotroblastic cell invading or penetrating this stromal lining, what is happening? They goes inside more and more. And how this process occurs? Because this syncytiotroblastic cell releases some hydrolytic enzyme, and as this releases some hydrolytic enzyme, they start breaking stromal cell from the endometrial lining, and as this goes more and more, what is happening? Another step begins, that is called the penetration and adhesion. The penetration and the invasion. So by the release of hydrolytic enzyme from the syncytiotroblastic cell, they start removing this stromal cell and replacing this structure inside this endometrial lining. So for better understanding, let me draw this large section here. So here is the blastocyst, and there it is, the outer lining, and the outermost lining of this blastocyst is the syncytioblastic cells. These are the multinucleated cells, and the inner one is the cytotrophoblast, which is mononucleated. So as the penetration or invasion begins, what is happening here is that this whole structure goes inside this endometrial lining and completely buried inside this endometrium. So here it is, the endometrial lining, and after complete invasion, this is the endometrial lining, that is the functional layer. What is happening? As the invasion completes, a fibrin clot develops here, and this closes the wall. So this type of invasion or the implantation here is called the interstitial implantation, and this is complete by the 10th or 11th day after the fertilization, in which the whole of this blastocyst goes buried inside this functional layer of the endometrium, and along with that these syncytioblastic cell start invading inside the maternal capillaries. So then these cells invading inside this capillaries, this syncytioblastic cells, and as these cells expand, they create spaces, and the space is called the lacunae, and finally these lacunae merge together to form the lacunar network, and slowly these maternal capillaries drain blood inside these lacunar network. So the maternal capillaries draining blood inside these lacunae. So along with this trophoblastic layer, these inner cell mass also differentiate themselves and forms the two layer, and these two layer called the bilaminar embryonic disc. Bi means two layer, so they form bilaminar embryonic disc. Here it is, the two layers, and this layer called the epiblast, and these epiblastic cells moves inside and cover this cytotrophoblast layer and forms a cavity within this is called amniotic cavity, and the hypoblastic cells again move inside and cover the cytotrophoblast layer and forms the cavity here is called the yolk sac. So this is how the complete invasion takes place, but some chemicals, some local peptides or cytokines are released from these syncytioblastic cell as well as the endometrial lining to stop further invading this blastocyst more deeply inside the myometrium as well as the perimetrium. So this is how we have studied how the implantation takes place, how this blastocyst, that is the ball like structure, goes inside this endometrial lining, that is the functional layer, and now onwards this endometrial lining is called decidua. So in my next part we will be seeing what is decidua and what are the layers of decidua and what is the function. Thank you.